Hello, hi, a very happy good evening. So this is Krishnaveni and we've been talking about the story of life on earth, and that's biology. Yes, welcome to my session. So this is lecture 2 of molecular basis of inheritance. So this is a very interesting topic that we started off. We were talking about the structure of DNA. So today we'll move forward and talk about certain experiments which prove that DNA is the genetic material. And if time permits, we'll start off with the first step in your central dogma of life, that is replication. So this is the agenda of today's lecture. So with this information I move forward. So thank you so much for watching my lecture today. So please don't forget to like, share and subscribe. Okay, so without any further delay, so let's move forward with the molecular basis of inheritance. So this is lecture 2. Okay, so shall I start? So here we go. So before we go into today's lecture, so I want to give you a small announcement. So today once again we are meeting at 6 p.m. and talking about the previous year questions, and we are talking about the year 2019. So we were discussing class 12 questions. So from there we'll move forward and finish 2019 if possible today. Okay, yes, Anthony Abhi, hello, hi, a very happy good evening. So thank you for joining my lecture. So please do like, share and subscribe. So that's okay, Anthony Abhi, that's completely fine. So please do join me at 6 p.m. as well. So we are discussing the previous year questions. Yes, so this lecture is at 6 p.m. Right, so please don't forget to join for this PYQ series at 6 p.m. Yes, Anthony, I'm perfectly fine, thank you. Yes, sir, a very happy good evening. So thank you for joining my lecture today. So please do like, share and subscribe. Yes, Satyendra, hello, hi. So good evening and thank you so much for joining my lecture. So it's raining here today. So yesterday you told me it was raining in your place. So it's raining very heavily in Kota. So it was like a thunderstorm was happening, so things were falling off the place. So it was a rain with a lot of sound, a lot of breeze as well. So as such, the rain was less, the breeze was more. But yes, it is a rainy evening in Kota. Okay, yes, so here we move into today's lecture, that is molecular basis of inheritance. This is lecture 2. Okay, so here we go. So what were we discussing in the previous lecture? So we spoke about the DNA, the genetic material. So DNA stands for deoxyribonucleic acid. So today we will talk about the search for the genetic material. So how did we arrive at the conclusion that DNA is the genetic material? Okay, so it's the same there. Okay, in your place also it's raining. Okay, so here also it's raining heavily. Yes, Shimpi, hello, hi, a very happy good evening. Yes, I'm good, thank you. How are you? Hi Prince, yes, a very happy good evening. So thank you guys for joining my lecture today. So please don't forget to like, share and subscribe. Okay, so it was literally raining that I had my legs and my completely wet the other part, which is not visible here. Yes, it was raining pretty heavily here. Alright, so it's sometimes good because it was really very hot in the morning. Very hot that you couldn't stand out without the AC. You start sweating so much that you drench. So it was that bad, it was that so hot in the morning. Yes, this now rain made me feel much better. Yes, Keshav, I'm good, thank you. So you're half asleep, that is well understood. Yes, okay, so please wake up, come on, quick, go wash your face and come. Okay, so here we move forward. So today we're going to talk about the search for the genetic material. Okay, so then we are going to talk about the RNA border. So it's near Bhopal, Nepal border. Okay, fine. Okay, well, I am in Kota, so it's raining in Kota. Fine. So yes, RNA world. So we'll talk about how dominant your RNA world is, and if time permits, we'll start off with the process of replication. Right, so here we go, a quick recap of what I was telling you in the previous lecture. So we were talking about DNA. So DNA is a nucleic acid. So it is made up of polynucleotides. So we have 4 nucleotides. One is adenylate acid, the other one is guanylic acid, then you have thymidyllic acid, then you have cytidyllic acid. Yes, so these are the 4 nucleotides that is present in your DNA. So it's raining in Shimpi's place as well. Okay, in Canada it is 121 degrees Celsius, so which means it is pretty hot. So on an average it is 50 degrees in Canada, that is 49.5. So yes, that is also pretty hot. Yes, I heard. So Canada is also drenching itself in the scorching sun. But yes, Kota was also pretty hot, but now it's fine because just now it rained. Okay, so the problem is that when it rains, no, you start feeling more hungry because your metabolism becomes pretty quick because the outside atmosphere and the temperature comes down, right? So that is the problem when it rains. So when it rains, all that comes to my mind is the beautiful shower, the beautiful water that you see, and hot things that you can eat, that is hot samosas, hot pakoras, chai. That's what comes into my mind, because when it's raining and when the weather is pleasant outside, your body's metabolism happens very quickly that you tend to feel more hungry. So if you see an individual in the summers, in the rainy season, he eats more, his appetite a bit increases compared to the hot summer season. Okay, yes, so this is about your DNA, the polynucleotides. So we go from here. So nucleotides are made up of your nitrogenous base, your sugar. So these two are bonded by glycosidic bond, and this two makes up your nucleoside. Okay, so then you have your phosphate group. So when your nucleoside binds to the phosphate group, so this binds via your phosphoester bond, and this forms the nucleotide. So in your nucleotide, if this is your nucleotide, the 5' end has phosphate moiety and the 3' end has your OH group. So nucleotides are added to the 3' end. Okay, yes. Shimpi says only next problem. Okay, fine. So you're saying momos? Yes, momos are also very good if it's very hot. Okay, so now don't tempt all of us. So then everyone will become hungry, including me, Keshav and Anthony Abhi. So Keshav is asking, Anthony Abhi, are you in Kerala now? Okay, fine. So digestion and absorption kab hoga? Okay, so I guess you're new to my session. So Monday, Wednesday, Friday, I teach a class 12 topic. Tuesday, Thursday, Saturday, I teach a class 11 topic. So yesterday was Tuesday, I started up the topic. So I will continue that on Thursday. Okay, Ranjan? Yes? Is that fine? Yes? Okay, fine. So this is your nucleotide, and this is what we were talking. So yes, hi Priyanka, yes, a very happy good evening. So what a surprise, you're here. So thank you for joining my lecture. So please do like, share and subscribe. So this is your nucleotide. So totally how many base pairs in humans? So the base pairs is a pair of nucleotide. So humans are diploid. So we have 6.6 into 10 to the power 9 base pairs, right? So this is the amount of base pairs that we have. So here we also spoke about who gave the double helix model. That was your James Watson and Francis Crick, who gave us the double helical model of DNA, right? So what is so important about this double helical model of DNA? What is the hallmark of this double helical structure is that you have complementary base pairing. So if I have one strand here, that is 5' to 3', the other strand has complementary base pairs. So A pairs with T, so A pairs with T, T with A, G with C and C with G, right? So this is complementary base pairing. So A binds with T using a hydrogen bond. So here it is 2 hydrogen bond. So G binds to C with 3 hydrogen bond. Okay, yes Priyanka, thank you. So this is how it is. So this complementary base pairing is the hallmark of your double helical DNA. So then we spoke about your Erwin-Chargaff's rule. So Erwin-Chargaff's rule is valid only for double-stranded DNA. So this is valid only for double-stranded DNA, okay? So what this says, so the amount of A is equal to the amount of T divided by the amount of G is equal to C, which is a constant or which is 1. So this is your Erwin-Chargaff's rule, okay? Okay, you go near City Mall to have momos. Okay, fine. So you miss Kota, alright. So this is your Erwin-Chargaff's rule, and this stacking of the DNA one over the other confers additional stability. Clear? Yes. And the distance between these two bases is 0.34 nanometer, or yes, the distance is 0.34 nanometer. So one turn or one side of the DNA, it has 10 base pairs, and the height of this, that is pitch, is 0.34 nanometer. Okay? Fine. So then we spoke about the sugar of your DNA. So in your DNA, which has the sugar as deoxyribose, it is a pentose sugar, right? So first carbon, second carbon, third carbon. So this third carbon has free OH group and it was ready to bind to the next nucleotide. Right, so this is 4 and this is 5. So this is attached to the phosphate group, and here I have my nitrogenous base. Right, so this is in case of your DNA. How about in case of your RNA, which has your ribose sugar? So this is also a 5 carbon sugar. The first carbon, the second carbon, third carbon. So the third carbon has a OH like your DNA. Okay. The second carbon also has a OH. So this OH group at the second prime carbon, okay, so this makes the RNA very, very reactive, thus decreases the stability. Okay, done. So this is what I discussed in the previous lecture. So today we'll move forward and talk about the search of your genetic material. Okay, so discovery of nuclein by Michener. So what is nuclein? Yes, it makes it more reactive, thus it is less stable. Okay, so what is nuclein? So nuclein, the term nuclein was coined by Friedrich Michener. So he said that DNA is acidic and it is placed inside the nucleus, and he called this as nuclein. Okay, yes. They have OH group, but the name does not end with O all. No, it does not end with O all, okay, because it is a sugar. It ends with OH, okay. So it is a sugar, it is not a compound. So this is a sugar, and here it is bound with a nitrogenous base as well as a phosphate. So its name does not end with O all, okay. So please don't bring chemistry into biology, okay. Yes, fine. So this was what Friedrich Michener did. So simultaneously you had the principles of inheritance by your Gerhard George Mendel. So both were simultaneously found at the same time. Okay, so yes, nuclein was discovered. So nuclein is what? Your DNA present inside the nucleus, and principles of inheritance, that is genes. So Mendel called them as discrete factors or genes. So they were also discovered at the same time, but still we could not find out which is the genetic material. Okay. So the discovery that DNA acts as a genetic material took long to be discovered. Why? What was the problem? So by 1926, the quest to determine the mechanism of genetic inheritance reached the molecular level. So we want to find out at the molecular level which is getting passed on. So genes, where are they present? On the DNA or on the RNA? Okay, so which molecule was actually the genetic material? So this question was taking the life out of the scientists, so they wanted to find out what is the answer. Okay, so this quest led to a series of experiments. The first experiment is your transforming principle. So please, you have a set of 3 to 4 experiments, basically 4 experiments in total. So I want you to remember the name of the scientist, the name of the experiment, the name of the strain that is used, and the conclusion. So these 4 points in each of the experiment is absolutely important. So these experiments were performed to find out that DNA is your genetic material. Okay, so this experiment name is transforming principle. So this is the name of the experiment. This is the first experiment. Okay, so number 2, you have to remember the name of the scientist. So the scientist here is Friedrich Griffith. Okay. So he was the scientist, and what is the strain that he used? So he worked on Streptococcus pneumoniae. So Streptococcus pneumoniae is a bacterium. Okay, so this is a bacterium that causes pneumonial infection. So there are two strains of this bacterium. So one is R strain, the other one is the S strain. So R strain because it has a rough polysaccharide coat. So bacteria has a polysaccharide coat. Okay, so this Streptococcus pneumoniae had two types of strain. One is R strain, that is rough strain. Okay, so this rough strain means it had a rough polysaccharide coat and it was less virulent. Okay, so S stands for a smooth polysaccharide coat. So when this bacteria is covered by a smooth polysaccharide coat, so it can easily bypass the immune system of the mice or the human. Okay, so that is the reason this is smooth. So it has the smooth polysaccharide, so which makes it easily to escape from the immune system of the humans or the mice. Okay, so this is the S strain or the smooth strain, and this is highly virulent. Fine, is the introduction clear to everyone? Yes, yes Dharani, a very happy good evening. So thank you for joining my lecture. Hi Swami, yes, good evening. So thank you for joining my lecture. So please do like, share and subscribe. So is this clear to everyone? Yes, this part is clear, no doubt. Fine. So what did Frederick Griffith do? So Frederick Griffith took out the rough strain, that is the R strain, and injected into the mice. So the mice lived as usual. The mice did not have any problem because the immune system is able to detect this bacteria and kill them. So then he took a smooth strain, that is S strain, which is virulent. Virulent means it is pathogenic. Okay. So when he injected that into the mouse died. Okay, so this is one part. So the next one, he took a heat-killed S strain. So he had this S strain, so he heated it. So when he heated it at high temperature, the bacteria died. So then he injected into the mouse surprisingly lived. The mouse was alive. Okay. The next one, he combined the rough strain, that is R strain, and the heat-killed smooth strain, and he injected into the mice and the mouse died. So how is this possible? So I told you this is non-virulent, this is virulent. So if you give this inside the mice lived. If you gave this, the mice died. So when you heated this S strain, the mouse lived, meaning the bacteria is dead. So it did not actually cause any infection. Okay, but when he mixed the R strain with the heat-killed S strain, then the the mouse died. So how is that? Yes, 2 mouse died. Yes, that is really sad. Okay, so why? What is happening? So the conclusion that Griffith gave was, so something from the S strain. So something from the heat-killed S strain, it got transformed into the R strain. That killed the mice. So this was his conclusion. Okay, but what got transformed? If it's proteins that went inside or that got transformed, if it was RNA or if it was DNA, so we did not know. So this was his conclusion. So this was the first experiment that Frederick Griffith worked on. Yes, is this clear to everyone? Yes, no doubt. So can I move forward? Yes. So please remember the name of the experiment, the name of the scientist, the strain that he used, and the conclusion that he gave. Yes, Shimpi, is it clear? All the doubts cleared? Okay, so I move forward to the next part. Okay, so still the answer is midway, right? So we do not know what transformed, right? So thus we have the second experiment in line. So the second experiment is the biochemical characterization of the transforming principle. So this is the second experiment. So Frederick Griff did not take up the mouse that died at the end and find out what actually transformed. He stopped there. Okay? Yes. So there were another group of scientists. You have 3 scientists. Okay? So the scientist's name is Avery, McLeod and McCarthy. So there were 3 scientists who did the biochemical characterization of the mice which died by the transforming principle. So what they did was, so they took the rough strain, okay, so the R strain plus the heat killed S strain, right, and they have put it inside the mice. So according to Frederick Griff, this mouse died, right? Yes? How? That is what they have to find. So in this experiment, first they gave proteases. So proteases breaks down the proteins in the bacteria. So if proteins are broken down, they checked if the mice still survived or the mice died. So when they gave proteases, the mice died. Okay? Next, they gave RNases. So they killed all the RNA in the bacteria and checked if it can still transform into the mice. Yes, it still transformed. So when they gave DNAs, that is when they broke down the DNAs, this transformation did not happen and the mice lived. So thus they found out it is DNA, that is the transforming principle, that has killed the mice. Okay, so this was the first logical conclusion that was arrived. Clear? Yes, so this is the second experiment. So these are the scientists, they worked on the transforming principle strains itself. The conclusion was that DNA was the transforming principle. Done? Yes, clear everyone? Okay, so now whatever I have explained, it's given in my slide. So initially they thought the genetic material was protein, but then you had Oswald Avery, Colin McLeod and Macklin McCarthy. So these 3 people came and they continued their work on the transforming principle in Griff's experiment. So DNA alone from the S bacteria caused the R bacteria to become transformed. So this was their conclusion. Okay. So 2 experiments are done. Yes. So now we go on to the third experiment. Okay. So genetic material is DNA. Who gave the final conclusion? So that we will see. So this was your Hershey-Chase experiment. So he gave the final conclusion, or they gave a commendable evidence that DNA is your genetic material. So let's see what this experiment says. So this is experiment number 3. Okay. So the name of the experiment itself is Hershey-Chase experiment. Okay. Right. So of course the scientist is pretty clear. So it is Hershey and Chase. So please remember the name. So what is the experiment that they did? So they took a bacteriophage. So bacteriophage is a virus that infects your bacteria, right? So this bacteriophage, so they took 2 different types of bacteriophage. In one bacteriophage, they radio-labeled sulfur. So sulfur is not present in your DNA, but sulfur is present in your proteins. Okay? So they took another bacteriophage and they radio-labeled phosphor. So phosphor is present in your DNA, but it is not present in your protein. So we have 2 bacteriophages, each with a selectively radioactive compound. So how does a bacteriophage infect? So bacteriophage is a virus. So at the end of the day, it's going to integrate its genetic material inside the host organism, right? So this bacteriophage, these bacteriophages were allowed to infect your bacteria, okay? So which means the genetic material enters inside the bacteria. So this is infection, okay? Now the genetic material gets integrated with the genetic material of the host, that is the bacteria. So the genetic materials blend. So that is number 2. Once they have blended with them. Okay, once they have blended together, then you do centrifugation. So centrifugation is high speed separation of the compounds. Okay, so if I have a bacterial cell, if I subject it to centrifugation, that is at low temperature and high speed, then the components settle down. So from that I can isolate DNA, protein and shape. So when I isolated, I only found radio labeled phosphorus, but I did not find radio labeled sulfur. So at the end I found only radio labeled sulfur, which means only the DNA has been integrated inside the bacteria by this bacteriophage. Okay, so the radio labeled sulfur was not present. So thus the conclusion was that DNA was the genetic material that can be passed down from one generation to the other generation. Yes, is it clear? Yes. Hershey's is choco chips, ma'am. Okay, so Hershey's syrup also we have, right? Yes. Yes, Hershey's syrup also we can have. So please try this. So take bread, take cheese, add Hershey's syrup. So it has the best combination. So Hershey cheese you can remember, or Hershey chase is the experiment. Alright, yes, fine. So this is the third experiment, that is Hershey's chase experiment. So can I move forward? Yes. Okay, so here I move forward. So you guys are not liking my session, but you're disliking my session. Huh? Okay, so properties of a genetic material. So now I have come to a conclusion that DNA is the genetic material. Now there should be some criteria that it fits that DNA becomes my genetic material, right? Yes, you have cleared NEET, okay? So you have cleared the NEET exam, that's fine. But for you to enter into a medical college, you should also have some prerequisite that you should belong to this caste, you should be from the non-creamy layer, you should have 75% of your 12th marks. Yes, so and so criteria you have. So let's see if certain criteria for being a genetic material it has. Okay, yes. So it should be able to generate its replica. Okay, so meaning it should be able to replicate itself. So if there is a DNA, only if it replicates, it can move from one generation to the other generation. Yes, I know, so you people won't do that, okay? So it should, so that's okay, that's fine. It's not that everyone should like my video. Yes, there can be critics, so critics make me more better. So they make sure that I improve, that I keep improving. That's fine. So it should be able to generate its replica. So if you have a genetic material, it should be able to replicate. So that is why when a mother cell divides, it can give two equal daughter cells. Okay. It should be stable chemically and structurally. It should not be reactive with all the reactions. Okay. So that is the problem. It should be stable chemically and structurally. Your RNA is not stable chemically and structurally because it has a OH group at the 2' position, which makes it very reactive. Any reaction it takes place, your RNA is vulnerable. Okay. So they provide scope for slow changes, that is mutation, which leads to evolution. So if you see your coronavirus, yes. So I hope all of you know coronavirus is the causative agent and COVID-19 is the infection. So please say COVID-19 as an infection, the causative agent is coronavirus. So the genetic material of your coronavirus is single-stranded RNA, just like your HIV virus, right? So this RNA is very prone to mutation because it is as such very reactive because of the 2' OH, right? So it is prone to high rate of mutation. That is why in a very short span, you had so many variations or mutations of this virus. If it had been a DNA virus, then the mutation would have been pretty less. So by now we have seen 4-5 mutations of this, which is very virulent, right? So that is because of the RNA, the genetic material that is present. So able to express itself in the form of Mendelian characters, that is genes. Genes have a pair of alleles. So it should be able to replicate itself. Yes, even RNA can replicate itself, but RNA is not stable chemically and structurally. So here only DNA is perfectly fitting. Then it should provide scope for mutation at a slow rate. So once again DNA fits this. So only DNA is able to express itself in the form of Mendelian characters. So thus the final conclusion was DNA is your genetic material. Right? Clear? Okay, so moving forward, talking about your RNA world. So though RNA did not fit all the criteria of being a genetic material, so RNA was the ancient nucleic acid that was ever discovered. So this was the ancient genetic material. Okay, so this was discovered way before your DNA, but gradually it gave birth to mutations at a high rate. But from your RNA, your DNA evolved. So over evolution, your RNA gave birth to DNA. Okay, but RNA world is more dominant. So this will once again be proved in the process of transcription, especially in the post-transcriptional step. So where does this transcription come? So this comes in a particular process known as central dogma of life. So clear? So this is about the RNA world. Okay. So now talking about the process of DNA replication. So we have recently completed a chapter of cell cycle and cell division, which says your DNA replicates in the S phase. Right? S phase of the interphase. How does it replicate? So it is not an easy process. It is a complicated process which happens. So here we talk about the first step of replication. So before we go into that, what is central dogma of life? So central dogma of life. So dogma is a set of principles that is validated, that is made sure it is true. Okay. So this central dogma of life was given by Francis Crick, the person who discovered the double helical model of DNA. Okay, so this Francis Crick, he told us the central dogma of life is that, so the information from the DNA is copied onto the mRNA, and from the mRNA it is translated into proteins. So this is the central dogma of life. First, DNA replicates itself so that it can be divided into the daughter cells, and then the information on the DNA is copied to the mRNA, and then it's translated into proteins. So your DNA is first replicated. So the process of copying the information on DNA into the RNA is known as transcription. Okay. And the process of converting your mRNA or translating into proteins, it is known as translation. So he called this as the central dogma of life. So this was given by Francis Crick. So in every cell has DNA in your body, right? So each of the DNA undergoes this process in a regulated way. So first we'll talk about this process of replication, then we'll move into the process of transcription and then translation. So now welcome to the process of replication. So DNA replication, how it happens. So the scientists ensured that DNA replication is semi-conservative mode of replication. So what is this semi-conservative mode of replication? So my DNA is double-stranded, right? So let's go to your favorite blackboard. So DNA is double-stranded, right? It is like this. And for your kind information, DNA has antiparallel polarity. So if it's 5' here, this is 3'. 3' and 5', right? So if this is my DNA, if this has to replicate. So what is replication? So what do you understand by the term replication? So replication is nothing but producing copies of itself. or you can say it is the process of duplicating. Right? So how does it happen? So if I have two strands, right, so first these two strands separate from each other, 5 prime, 3 prime. So they separate quite apart from each other. Then the new strand is synthesized. So this is my new strand, right? So this is the new strand. So now here again the new strand is synthesized. So if you see, this is one strand, this is one double helical strand, right? So each of these strands, they have one parental strand retained. So one double helical DNA can in turn replicate to give me two separate strands, right? In each of the strands, one of the parental strands is retained. So this is my parental strand, this is my parental strand. So again, if this replicates, the parental strand will be retained. So since each of the new strand retains a parental strand, so this mode of replication is known as semi-conservative mode of DNA replication. So you understood? Yes. So how is the DNA replication? It is semi-conservative because in each of the new strand, a parental strand is getting replaced, or a parental strand remains in each of the new strand. Is this clear? Yes. So now I will tell you how this replication happens. So here easily I drew a line, but we are going to talk about the entire process of replication. So this is clear? Yes. Fine. So who proved this semi-conservative DNA? So at the end of the day, we need proof, right? So if you tell me you have studied, how do I believe? So that's why I keep Menti, right? So how do I believe? Yes, DNA follows semi-conservative mode. So this experiment was proved by another set of scientists. So this is for your DNA replication. So the name of the experiment is semi-conservative DNA replication. The name of the scientist is Michelson and Stars. It is mentioned here. Michelson and Stars experiment. Okay. So how did they do? They took a double-stranded DNA. So this DNA has your nitrogenous base, right? Because it is a polymer of nucleotide. So which means it has nitrogenous base. Nitrogenous base consists of your nitrogenous compound. So here the parental DNA had N15 nitrogen. So he put this parental DNA containing N15 nitrogen in a media containing N14 nitrogen. Okay, so here he gave N14 nitrogen media. Okay, yes. So after 20 minutes, so 20 minutes is the time that is needed to replicate. Okay, so after 20 minutes, when it became two new strands, he found that one strand retained N15, that is my parental strand, and the new strand was N14 because the medium was N14. So here also he had N15 and here also he had one N14. Okay, this is first 20 minutes. After 40 minutes, when he had 4 strands, again he found out that each strand has one parental strand and one new strand. Thus, he came to a conclusion that DNA undergoes semi-conservative mode of replication. So, this is experiment number 4, but this is for your replication process. So, now quickly, I will tell you how the process of replication happens, but before that, if E. coli was allowed to grow for 80 minutes, so one strand divides in 20 minutes, if I am given 80 minutes, that is 3 divisions can happen, then what would be the proportion of light and hybrid densities in the DNA? So, in every division, I have one parental strand retained, right? So, in the first strand, in the first division, I have 2 parental strands retained. Okay. If I go for the second division, again I will have, so So here I have one parental strand, here I have one parental strand. So in each division I have two parental strands retained. So this is my first division. I have one parental strand, two. Now this is my second division. If you see, here I have one parental strand and one parental strand. So in each division, the first parental strands will be retained. So it will be two heavy density and the remaining will be the light density ones. Okay, so N14 is light, N15 is the hybrid, and N15 is the heavy isotope. So in each division my hybrid will increase, but my parental strand will remain as 2. Okay. So similar experiments, they used radioactive thymidine instead of nitrogen to detect the distribution of the newly synthesized DNA in a chromosome. But this radioactive thymidine was performed in your Visia faba. So it is your faba beans and it was done by the scientist Taylor. So please remember this name. So he also proved that DNA in chromosomes replicate in a semi-conservative mode. Okay, so now let's quickly talk about how the process of replication happens. Okay, so ready? The process of DNA replication. So how long is your DNA? Yes, so your DNA is 2.2 meters long, right? Yes, I know your DNA is double helical structure, but for my understanding, I will draw it as a linear structure. Suppose if this entire 2.2 meter has to replicate, okay, so I told you the two strands separate, right? So this entire 2.2 meter cannot separate from each other because it will cause destability to the DNA. So at a stretch, it will not unwind throughout the entire length of DNA, but it will unwind at a certain portion where it is about to replicate. So if this is my DNA, so it will unwind at only one portion. So it's going to replicate in this direction, but it will unwind in bits and portions. Okay, so the place where it unwinds, so this is known as your replication fork. So this is your replication fork, this and this is your replication fork, this point and this point, okay. So this bubble where your replication is going to begin, so this is known as your replication bubble. Okay. So your DNA will not unwind throughout the length because that will use up a lot of energy and it will cause destability to the DNA molecule. So it happens in a small portion. Okay. So now I am talking about this portion. Okay. I am just explaining to you. So suppose let me draw it like this for my understanding. So DNA has antiparallel polarity. So it is like this. Right. So this is my replication fork. This is my direction. Fine. Now, who will unwind the DNA? So, will I go pick up my DNA and unwind it like this and give? No, right? So, everything has to be done automatically. So, to unwind the DNA, to initiate the replication, so the important players are your enzymes. Okay? So, the one which unwinds the DNA is an enzyme. So, what unwinds the DNA? That is your helicase. So, helicase unwinds the DNA. Okay? Yes. So, now after your DNA is unwind, so you have some stress. So, if something is tied together so tightly, if you pull them apart, they again have some kind of tension. So, if you have rubber band, okay, if you release the rubber band, it has some tension that it wants to relieve, right? So, imagine you have two springs which are tightly attached together. You pull them apart with a lot of force because they try to go back to their original position, right? So, once you unwind the DNA, there is some kind of stress on the surface. That stress is relieved by your topoisomerase. So that stress is released by your topoisomerase. Fine? Yes? Clear? Done. Now, this is open. So from where exactly the process of replication is going to begin? So how do you know where exactly it is going to begin? Okay. So it's going to begin at a particular point, okay? It knows where to begin. So that particular point where your process of replication starts, that is known as the ORI site. That is origin of replication. Okay, but who is going to do this origin of replication? So you have another enzyme, that is your DNA polymerase. But hold on. This DNA polymerase is the most important player because it is going to polymerize your DNA. Okay? Yes. So this DNA polymerase has 2 points. It can only polymerize DNA in the 5' to the 3' direction. Okay? And it cannot start the process of replication. It can only polymerize it. It has a starting trouble. So then how is it going to happen? So imagine like this. I have beads with me. I have a string. I have to put the beads inside the string. Okay, but my DNA polymerase does not know how to put the first bead inside. So if someone guides me, this is how I have to put in, then my DNA polymerase can easily put everything. So I have my beads here, I have my string. Okay, so now I have to take the beads one by one in order and put it inside the string. So that is the function of DNA polymerase. So DNA polymerase can put the beads in particular order, that is from 5' to the 3' end. Okay, but it does not know how to put the first bead inside. So someone has to initiate this process. So who's going to initiate this process? So you have another enzyme that is known as your primase. So this primase produces a primer. So what is a primer? So primer is a short oligonucleotide sequence. So it is a short oligonucleotide sequence. So oligonucleotide means it has less than 10 nucleotides, and these nucleotides are complementary to the template strand. These are complementary to the template strand. So what is the template strand? This is the template strand, right? Yes. So I have a small primer, okay? So this has complementary sequence to the template. So it goes and attaches properly, right? Fine. Now the DNA polymerase now can easily synthesize the DNA. It can synthesize from the 5' to the 3' end. So the primase synthesizes primer, which is the short oligonucleotide sequence, which goes and binds to the corresponding sequence in the template, and now your DNA polymerase can initiate polymerization. Okay, so DNA polymerase can synthesize or polymerize in the 5' to the 3' direction. But this strand has antiparallel polarity. So how is it going to synthesize here? So here it will synthesize in bits and pieces like this, because here the 5' end is towards this end, right? So it has to synthesize in the reverse direction. So it will synthesize in bits and pieces, and these fragments are known as Okazaki fragments. So later these Okazaki fragments are joined together. So later these fragments will be joined together by your DNA ligase. So the Okazaki fragments are joined together by DNA ligase. Okay? Yes. So this strand, which is synthesized continuously, is known as continuous strand, or it is known as your leading strand. So this one, which is synthesized in bits and pieces, is known as the lagging strand or the discontinuous strand. So this is the leading strand, this is the lagging strand. So this is the discontinuous strand, this is the continuous strand. So now everyone got how the process of replication happens? Yes? Yes? So there is a small twist to this. So let me see if you can understand the twist. So I have my base pair DNA like this, 5 prime to 3 prime, 3 prime to 5 prime, right? So how does the process of replication have? So this is separated and this is separated, right? Suppose if I have the sequence G, A, T, T, C, C, G. Okay, yes, my DNA polymerase, my primase comes and bind here, and my DNA polymerase will synthesize this strand. How it will synthesize? How it will synthesize? So this will be complementary C, G, G, A, T, C, right? So your DNA polymerase will only pick up C and put it here. Your DNA polymerase will not synthesize C, G, G, A. It will only pick up the beads and put it inside. The nucleotides are synthesized by your cell. So you have your purine and pyrimidine metabolism by which your cell can make its own nucleotides. The nucleotides are here ready, okay? So your DNA polymerase just picks up the correct one and puts it in place. So these are the substrate. So these nucleotides act as your substrate. Your DNA polymerase is the enzyme. Enzyme needs a substrate, right? So these nucleotides are high energy compounds like your adenosine triphosphate. Okay, so that is a nucleotide, right? So this provides energy for the process of replication as well as it serves as a substrate. Yes, to unwind the strand for your DNA polymerase to work, you need energy in the form of ATP molecules. Okay, so this energy is supplied by the substrate for your DNA polymerase. The substrates are your nucleotides. Your DNA polymerase just picks up your adenylenic acid and puts it in line. Okay, so that is only the function of DNA polymerase. Your DNA polymerase does not synthesize CGGA. Okay, here I have kept CAG. So DNA polymerase perfectly identifies it and puts it in place. Clear? Yes? Is this much understood by everyone? So no doubt? Clear? The process of replication? Yes? So I will continue. So we'll just read what is in the slides of replication in the next lecture, and in the next lecture I will start off with the process of transcription. So transcription will be on Friday. So today is Wednesday. So Friday we'll talk about the process of transcription. So please don't forget to join my lecture at 6 p.m. So we are going to discuss the previous year question paper. So we are going to discuss the paper 2019. So please join me for the same. And tomorrow at 4 p.m. we are continuing your digestion and absorption. So thank you, and this is me wishing you more and most. If you genuinely like my teaching, so please do like, share and subscribe. So looking forward to meet you at 6 p.m. So until then, stay tuned, take care and bye.